Hello everyone. So in this video of chapter electrochemistry, I'll be talking about quantitative aspects of electrolysis, then galvanic cell and voltaic cell. Let's get started. Quantitative aspects of electrolysis. We all know during electrolysis two half reaction used to happen. One half reaction is oxidation, another half reaction is reduction. The mass of reactant consumed or the mass of reactant produced at the electrode can be calculated by knowing their stoichiometry of the half reaction which is happening at a particular electrode. So if we want to calculate the quantity of electricity passed, we can find out with this formula Q is equal to I multiplied by T. I is current passed, which is measured in ampere, and T is the time measured in seconds. If we want to calculate the number of moles of electron passed, so for calculating this, total charge passed, which is Q, will be calculated with this above formula, and the charge of 1 mole of electron is 96,500 coulombs. It is also referred as 1 Faraday. So we can say here moles of electron passed is equal to Q upon 96500, and this would be the formula for calculating moles of electrons actually passed. Third thing which we need to determine is the calculation of moles of the product formed. For this we require balanced chemical equation, and that balanced chemical equation must be the half reaction which is happening at the particular electrode. The stoichiometry of first half reaction indicates the moles of electrons passed and the moles of product formed. For example, if we are talking about this reaction where cupric ion is accepting electrons to turn up into copper metal. Here cupric ion is accepting 2 electrons for producing 1 mole of copper. So we can calculate the moles of product formed by the moles of copper here. The moles of electron actually passed by the given equation this, and by that mole ratio would be calculated as moles of product formed in the half reaction upon moles of electron required in the half reaction. Here 1 mole of copper is forming, that is why moles of product is 1 upon. For formation of 1 mole of copper we are requiring 2 electrons, that is why it is divided by 2. So the moles of product formed is equal to the moles of electrons actually passed, which is 1 upon 2 we have calculated, multiplied by this is the mole ratio, and the moles of electrons actually passed would be by this formula, that is Q upon 96500. So if we are elaborating Q also, the Q is equal to I multiplied by T upon 96500 multiplied by mole ratio would be the formula for calculating mole ratio. So if we are going to calculate mass of the product, it would be calculated as W is equal to moles of product multiplied by molar mass of the product. Moles of product in the previous slide we have seen, its formula is IT upon 96500 multiplied by mole ratio, and if with this one is multiplied by the molar mass of product, will be given as W or mass of the product. Now suppose two cells containing different electrolytes are connected in series. Same quantity of electricity is also passing through them. The mass of the substances liberated at the electrodes of two cells are related as, suppose for one cell I am calculating W1 is Q upon 96,500 multiplied by its mole ratio multiplied by its molar mass. It is for one substance. Or I can also say Q upon 96500 Q upon 96500 is equal to W1 upon mole ratio of that substance multiplied by its molar mass. Similarly, if I am talking about the another substance, which is happening on the another electrode. So its mass would be W2, and I can say Q upon 96,500 is equal to W2 upon molar mass multiplied by mole ratio of the second compound. Here M1 and M2 are the molar masses of the substance produced by the electrodes of the cell 1 and 2. Because we know Q upon 96,500 will be the same value, so by cancelling them, or you can say by comparing them, we will get W1 upon mole ratio of the one substance multiplied by its molar mass is equal to W2 upon mole ratio of the second substance multiplied by its molar mass. Now galvanic cell or voltaic cell. Galvanic cell or voltaic cell are the cells where electricity is generated through the use of spontaneous chemical reactions. Now, a galvanic cell is made up of two half cells, and each half cell consists of metallic strip immersed in the solution of its own ions of known concentration. For example, a strip of zinc metal immersed in 1 molar aqueous solution of zinc ions form an half cell. It follows that two metal plates and solution of their ions with known concentration are required for the construction of a galvanic cell or voltaic cell. Two half cells are constructed by immersing two metal plates in solution of their respective ions placed in a separate containers. The two half cells so constructed are combined together to form galvanic cell. A metallic plate called electrode is connected through voltmeter by conducting wire for the transfer of electron between them, and to complete the circuit, the two solutions are connected by conducting medium through which cations and anions move from one compartment to another, and this requirement used to be fulfilled by salt bridge. This is one of the example of galvanic cell. Here there are two containers having electrolyte and electrodes. These two electrodes are connected via this conducting wire, and voltmeter is also attached to it. This conducting wire is necessary for flowing of electrons from one electrode to another electrode, that is from anode to cathode. Here these two solutions are connected by this U tube, which is inverted and immersed in both the solution. This U tube is called as salt bridge, and this salt bridge maintains the concentration of ions. Now, what do you mean by salt bridge? In galvanic cell, the two solutions are connected by salt bridge. It is U tube containing a saturated solution of an inert electrode such as potassium chloride or ammonium nitrate and 5% agar solution. The ions of electrolyte do not react with ions of electrode solutions or electrodes. Salt bridge is prepared by filling this U tube with hot concentrated solution of salt and agar-agar solution, and it is allowed to cool down. Then the cooled solution sets into gel, which does not come out from the tube even if we are inverting the tube, and the salt bridge is kept dipped in distilled water. Function of salt bridge. Function of salt bridges are: it provides an electrical contact between the two solutions and thereby completes the electrical circuit. It prevents mixing of two solutions, and it prevents, or you can say it maintains actually, electrical neutrality in both the solutions by transferring of ions. So these are the three main functions of salt bridge, which used to connect the two different electrolytes in a particular galvanic cell. Thank you.